Hi everyone, my name is Janica and I'm a Master of Research student from the Hawkesbury campus. This semester for experimental design and analysis, I completed a project to rapidly analyse antioxidant and phenolic compounds in native Australian food plants using techniques termed under post-column derivatization, which we'll call PCD, with high pressure liquid chromatography, or HPLC for short. In this presentation, I'll talk you through each of the techniques we used and all of the terms which may seem like jargon now to hopefully help each of you have a clear understanding of the work I did. The main reason I chose to focus on native Australian food plants, which we'll abbreviate to NAFs, and you'll find I like to abbreviate a lot of things in this presentation, but I digress, is because it is the main theme of my master's thesis. And one of the reasons that they're so interesting to me is because most of them contain large concentrations of bioactive compounds, or compounds with antioxidant, antibacterial, and anti-inflammatory, or etc. properties. Some previous work which validates this includes a study by Konsak et al. 2010, who found that Kakadu plum had 6 times higher total phenolic content than domesticated blueberries, and has the highest source of vitamin C of any known fruit in the world. And for those of you who don't know, phenols are a class of compounds which consist of a hydroxyl group bound to an aromatic ring. And the presence of phenols in NAFs indicates antioxidant potential, as all antioxidants are phenols, but not all phenols are antioxidants. In a similar study, Netzel et al. Found 6 of the 12 Australian fruits analysed in his study had a phenolic content up to 4 times higher than domesticated blueberries. And more related to this area of research, in a separate project, Konsak and his team found similarly high phenolic content in native Australian herbs and spices, namely Tasmanian pepperleaf, lemon myrtle, and anise myrtle, which had the higher antioxidant response attributed to cinnamonic acids and flavonoids. So why then, given all these antioxidants, have NAFs not been integrated into the Australian diet? And why are we, the Australian public, not educated on how to prepare or eat them? Well, these questions are what I plan to answer in my master's thesis, and may well extend into a possible PhD. But given that I only had a semester, I thought I should at least make a start. And so in this project, I plan to develop a more rapid alternative to accurately detect bioactivity in NAFs. In the previous studies I discussed, much of the methods used were labor-intensive and time-consuming. PCD presents a faster alternative detection method when coupled with HPLC, which is an instrument which separates out analytes or components of compounds within a column according to polarity in a liquid solvent, or called mobile phase, for detection and identification. Here, analytes that elute out of the column in the mobile phase can be mixed with a derivatizing agent, such as 2,2-diphenyl-1-picrohydrazole, or DPPH. This transforms each potential antioxidant analyte into a similar compound. This process is known as derivatization, and it is the response of this reaction which is detected to indicate potential bioactivity. So, in the case of determining antioxidant activity, we would use antioxidant-specific PCD reagents, such as DPPH and 2,2-azenobiz-3-ethyl benzoyl 6-sulfuric acid, sulfonic acid, or ABTS, as well as phenol-specific reagents, such as potassium ferrocyanide and 4-amino-antipyrine. Now, I know some of you are probably a little confused with what I said, but don't worry, in the next few slides I have diagrams and videos to help explain these techniques more clearly. What makes PCD assays possible is the use of specialized HPLC columns known as active flow technology, parallel segmented flow, or AFT-PSF columns. These columns feature a 3-component frit and a multi-port end fitting. The frit separates the wall and central flow of the mobile phase through an impermeable ring. This allows for multiplexing, or rather the connecting of multiple detectors, destructive or non-destructive, to each port for analysis. And here we have the real life example, and in a few more slides I'll show you how this column is connected in the multiplex mode and where each of the ports are connected to. Reaction flow, or RF mode, is a technique where the reagents are pumped against the direction of the mobile phase and mixed with the analytes in the peripheral port of the end piece. In doing this, reaction coils, as shown here, or coiled lines which help to equilibrate the mobile phase when mixed in with reagents, can be reduced in size or eliminated from use. This sharpens chromatographic peaks and increases the resolution and selectivity, as reaction coils can cause a loss of separation power and peak broadening through post-column flow deadening. And so, with that in mind, the aims of this study were to detect antioxidant and phenolic compounds in two NAFs, cinnamon and lemon myrtle leaves, abbreviated to CM and LM respectively, using the following HPLC-based techniques. 1. A multiplexed experiment with MS, UV-Vis, and DPPH detection using the AFT-PSF columns explained earlier. 2. Using ABTS to detect antioxidants using the AFT-PSF column in RF mode. And 3. The use of potassium ferrocyanide and 4-amino-antipyrine as a two-component PCD reagent to detect phenols, again using the AFT-PSF column in RF mode. Given the previous studies we discussed in the introductory slides, we expected to see a positive response from CM and LM extracts to each of the three PCD reagents used to detect antioxidant and phenolic compounds. As illustrated by this flow diagram, CM and LM leaves were pounded separately in a mortar and pestle. With Miller-Q water, the solution was then sonicated in brown sample vials for 5 minutes at room temperature, and then filtered through a 0.22 micrometer filter using a syringe into 1.5 milliliter HPLC vials, ready for the PCD experiments.
Okay, so on this slide, you'll see on the left-hand side we've got a schematic of the multiplex detection setup using the AFT-PSF column. And to help you, I guess, understand this concept a little bit, I thought we'll play this video so you can see what it looks like in real life as well. So at the moment, we're just walking through the multiplex setup and showing you all the different instruments. And we'll explain, I guess, how they're all connected as we go through. So at the moment you're just looking at the chromatogram for the DPPH detection. Here is the mass spectrometer. Okay, and then to the left here we have a pump for our sample extracts, and underneath that we have the sample tray and injector. Okay, and then over here we just have another pump which is not being used at the moment. The pumps for the DPPH. This is the sample tray for this HPLC system, which we're not using, so we won't worry about too much. Okay, and then we'll just come back here. Now I'm going to open this tray door so that you can see where the samples are being kept. So they're under refrigeration. I know it's a little hard to see, but each of those white dots there is one sample extract. Now that's injected through this injection port here into the column. Okay, and there's the column. And then you can see just at the end towards the right hand side the end piece. Okay, so as we described, the central flow will go into the mass spectrometer. So this is underivatized, so not mixing with any reagent, just as it is coming straight fresh from the column. We have one of the peripheral ports going to a UV-Vis vis detector. Again, this is underivatized mobile phase. Okay, and that's the UV vis detector. And then the other underivatized mobile phase is exiting out of the second port into a T piece to mix with the DPPH. And that is then the derivatized mobile phase going into a detector. So we have the pump down the bottom here and the detector at the top. Now the DPPH is covered in aluminium foil, just towards the left hand side of the screen because it's light sensitive, but you can see it through that purple coloured stream there that's exiting from it. Okay, so that's the DPPH. And that's the degasser, which takes any air bubbles from the DPPH out, because that obviously will influence the readings through the detector and it can cause a lot of problems for your column. Okay, and then the detector for DPPH will be picked up by this chromatogram here. And then coming back here, we have the UV vis and mass spec data.
Moving on to the other two PCD experiments carried out in RF mode, the first being the ABTS experiment, for which the setup is shown here in figure 4. Here the central underivatized flow is delivered straight to a diode array detector or DAD. Meanwhile, for ABTS to function as a free scavenging radical for antioxidants, it must first be mixed with potassium persulfate. So here both ABTS and potassium persulfate are mixed into a T-piece, and then the activated ABTS solution is pumped into one of the outer ports of the N-piece to mix with the analytes in the mobile phase. The derivatized flow is then sent from another peripheral port to the DAD. Moving on to figure 5, the phenol reagents potassium ferrocyanide and 4-amino-antipyrine are pumped into separate outer ports to mix directly with the peripheral flow, which is sent from the third port into a DAD. And like the ABTS setup, the central underivatized flow is delivered straight to the DAD. The observed UV vis responses for CM and LM extracts indicate compounds with high antioxidant activity elute early in the chromatographic separation. A total of 11 peaks were observed for CM and 12 for LM. MS data run in both positive and negative total ion current, or TIC, indicated few charged ions, or CIs, in positive mode and more CIs in negative mode. Similar results were observed for the MS data for LM, with greater relative abundance of CIs observed in negative mode than for CM. Interestingly, the single CI which appeared at greatest relative abundance in CM positive mode at 11.5 minutes was not present in the LM sample, suggesting the presence of a glucoside. Furthermore, the greater abundance of CIs in the negative MS spectra of CM and LM suggests that the antioxidants contained in these leaves are more likely to be acids such as chlorogenic acid rather than glycosidic compounds such as hesperotin rhamnoside. Moving on, of all the peaks for the CM UV vis chromatogram, only peaks 1 to 7 showed a positive response to DPPH, with peak 4 appearing the most potent. Comparatively, only peaks 1 and 2 in the LM extract expressed a positive response to DPPH, indicating less antioxidant potential than CM. The ABTS response of CM and LM illustrated similar chromatographic profiles to that of DPPH, which was to be expected as both are sterically hindered stable radicals. A total of 9 antioxidant peaks were observed in both CM and LM extracts for the ABTS assay. 1 peak in the CM and 3 peaks in the LM in the PR or phenol reagent chromatogram match in retention time to the peaks that were observed in DPPH and or ABTS assays, indicating that these peaks are more likely to be phenol-based antioxidants. To ensure the reliability of peaks observed in the PCD detection methods, CM and LM samples were also analyzed underivatized at the PCD analysis wavelengths. No significant peaks were observed at the PCD wavelengths, which confirms that the peaks observed in each of the PCD detection methods are derivatized peaks. Overall, in this study, we achieved our aims. We demonstrated that coupling HPLC with PCD techniques and multiplex detection was a more efficient method of screening antioxidant and phenolic compounds in native Australian food plants in comparison with other conventional techniques used previously. Time, as you can probably tell, was my biggest constraint in this project, especially since I only came into the project midway through the semester. Had we had more time, we could have looked at identifying the potential antioxidant and phenolic compounds detected, and we could have analyzed extracts prepared in solvents of decreasing polarity. Nonetheless, given the time available and our resources, I believe that this was a very productive 9 weeks. Given our current findings and constraints, I would hope to take this study to the next stage and actually identify compounds we detected using mass spectrometry or nuclear magnetic resonance. I would also like to prepare ethanolic extracts of both CM and LM to see if any other less polar bioactive compounds are detected. Sample preparation was relatively time consuming at about 1 hour per extract, so I would also like to consider different techniques to save on time. And finally, I'd like to look at other native bush foods, especially fruits such as sandpaper figs and lily-pilly. And here are the references used in the presentation today. I would like to acknowledge Andrew Shalika, Serkan Pravadali Kekik, and Andrew Jones for their immense support and help and kindness throughout this project. I would also like to thank Graham Jones for setting up this unit and allowing me to undertake a mini project in the first place, and to Erin Wilkins and the team at Murrumbidgee for their support and generosity in providing samples and an insight into Darug food culture and traditions. If there are any questions, I would love to hear from you, so please send me an email on the address below or submit your comment through VUWS.